Well, thank you very much, Martin. And of course, it's always a pleasure to be here. And if you look often enough, you'll find me bouncing back here quite often. And in fact, the real twisted tale of this was that Martin and I had the great fortune of getting a CFI grant to build an extension onto the Jack Bell Research Center, as you'll recognize here. And I think the real issue was that we couldn't decide whether to build on the east side or the west side of the building. So we decided to build on both sides. Martin's building a beautiful extension on the east side. I got the west side, and I think it was actually a zoning problem that we had that made me keep moving it a bit further west along the way there. And so, of course, when I came to the ocean, I had to keep on drifting southwest there. And really it's not my departure per se, but really it is the west wing of the prostate center here and carrying on that model. But more so actually creating what Martin was referring to is really this joint project between Martin and I to link the prostate cancer interest between Australia and Canada. And so this network that was recently funded is an open network. You're all invited to join it. It currently has about 200 scientists and clinicians in it between the two countries. And the point of that is to allow people to access each other's technologies and resources and come together in a collaborative spirit as we have in the Prostate Center, but doing this on a much more intercontinental scale. And so just contact me if you want more information on how to access that detail. So what I'm really here to do, though, today is to tell you a story that comes from the Prostate Center. And I set it out this way in a framework to show you that it's really a story that has come in a very integrated way from this discovery into the pipeline, and it's transversed its way through identifying and validating that the observations that we saw in high throughput models were were really key to the human pathology, of course, and validating that and then interlinking that with what's going on in clinical trials. But more importantly, this isn't just a linear pathway that goes from discovery to human testing. It has very important components of it, taking human samples from clinical trials and bringing them back in and trying to understand the pathways that are within those and the inherent resistant pathways in particular that might occur from a particular treatment. So this is the framework in which it's set. We're all familiar with prostate cancer progression as a disease, but my focus of my research has always been on the advanced disease side. And so trying to understand exactly what's happening upon that initial failure to tumors once you remove androgens. And so what is the molecular response beyond the cellular response? And trying to understand why it is that once those tumors have their androgens removed, of course, they have this initial response, but almost inevitably, as if it's a programmed and systematic change, if you will, along that spectrum, that these men will become what we used to call androgen independent, and that was always signaled, and still is, by rising PSA, of course, an androgen-regulated gene. Now, we know...
Sorry to interrupt from Colleen. We can't see any slides for some reason. Sorry about that.
Okay.
Are you seeing them now?
No, we can't see anything. We can just see shadows.
Me too. It's too early in the morning.
All right, bear with us. I'll get you to continue, and I'll see if I can work it out.
Okay.
Okay, thanks.
But we understand now a bit more that perhaps this end of the disease isn't really independent of androgens, and that's going to be largely the focus of what I'm going to tell you today. And this end-stage disease, which would have also been called hormone refractory, is now transitioning in its nomenclature to be called castrate-resistant prostate cancer, because truly that's what these men are resistant of is castration and that therapy itself. So to approach this, I mean, we've used a lot of different models, but my vision of this androgen independent or castrate-resistant prostate cancer progression is that it is really not a stochastic event. It's really a programmatic event that, in fact, is probably set up by castration itself. So there's an initial response to castration. It shuts off particular genes, turns on others. This allows for a particular nadir period in terms of what we see in following castration, but ultimately, of course, as we've said, this is going to result in this androgen-independent phenotype. So what I've been doing is looking at how genes change along this spectrum, and particularly trying to identify what would be a cascade of regulation that sets off, that ultimately creates this molecular phenotype. And part and parcel with that, I'll kind of skip to the point that in all of these cases, as we're looking at this, we see the reactivation of what would have been androgen-regulated genes. But really to show that and look at that in terms of patient samples beyond xenograft models, we've also taken advantage of the neoadjuvant hormone-treated patients, in which these men have had neoadjuvant hormone therapy prior to radical prostatectomy. We've laser captured out the tumor cells from this and performed the same type of high throughput gene expression analysis. And there's a couple of take-home messages to this, and that is that for particular panels of genes, untreated men are quite similar, and you'll see that treatment of hormone ablation, importantly, across these 6 men shown here, have a very similar response to that therapy in terms of having particular genes, shown in red, being turned on by androgen ablation, and having particular genes shut off. So there's a uniformity of response, and it's actually quite a robust response, both in time and magnitude. So this, to come back to trying to understand androgen action, of course, we know that androgens basically are signaled through having testosterone come into a prostate cancer cell. It's converted to dihydrotestosterone, binds the androgen receptor, and either activates or represses particular genes. Now, we've been trying to understand how does this process get dysregulated, and there's been a lot of hypotheses that are out there in the literature, certainly. And one of which is that, the androgen receptor can mutate and it can take on other steroids to activate, or it could be activated potentially having nothing to do with this steroid at all, and be activated by particular growth factors in kind of a subvertive pattern. But where we have come at this is wondering whether, in fact, androgens themselves are being made. And I'll tell you the rationale behind that. So when we look at what androgens do within a cell, and this is back in vitro, is that we can see androgens robustly, as shown here by R1881, a synthetic androgen, repress certain genes and activate other genes. And we've been trying to understand that not on a gene-by-gene basis, but really trying to pull that into pathways of activity biologically. And we can see then, if we look at this cellular pathway of these different genes, which these are transcription factors largely down here in the nucleus, and here's the cytoplasm and extracellular space, that androgens have a profound action to show that that this is actually a androgen receptor, though specific event. We can knock out the androgen receptor and then look to say what happens if we add androgens or remove them. And we can identify which genes are truly androgen and androgen receptor regulated. And that becomes important in how we integrate this data later. So one of these first androgen regulated genes that we looked at, and this was quite some time ago in 2004, really caught our attention because it was one of those master regulators that had the potential to activate in a very uniform way a variety of pathways. And there were pathways that were quite intriguing. And that is the pathways that are downstream of the activation of a sterol receptor element binding protein, which is a transcription factor, which we've shown to be androgen regulated, that controls effectively the whole entire synthesis pathway of fatty acids, as well as the synthesis pathway of cholesterol synthesis. And we began to wonder if it was also involved in steroidogenesis, as it is in other cell types. And so one thing to recognize in prostate cancer cells and prostates themselves is that they are one of the most lipogenic organs. They're more lipogenic, or as lipogenic as the liver, in fact. And this process, though, in the prostate is all under the control of androgens. So to look at this in vitro or within these tumor models, this is just showing that transcription factor following castration is shut off and it turns back on in androgen independent progression, as does all of the genes that are in the pathways that it controls. So there's kind of a uniform response of about 30 genes that are downstream of this transcription factor, as we would expect, but importantly, noting that all of these come back up or upregulated in androgen independent progression. And that's where, coming back to this theme, that in fact anything we see as androgen regulated is reactivated or re-repressed in some cases in androgen independence. And so to show that in terms of tissue samples, these are from humans, of course, and what we've seen is that this pathway, and this illustrated here by SREBP itself, is actually quite low expressed in relatively normal tissue or BPH. It's very elevated, in fact, in cancer compared to BPH. And following androgen ablation in these patients, you see this initial suppression of it, but then in androgen-independent tissues, you see these pathways reactivated. So this, of course, made us want to study it a bit further. But because it controls three major pathways, we've kind of dissected those out. And I'll only really talk in detail about one today, but it is to point out to you that if we block the fatty acid synthesis pathway, this actually induces an apoptotic response within prostate cancer cells. And we've continued to study down this pathway as to how that's happening and whether there will be a therapeutic approach in that regard. We've also looked at blocking cholesterol synthesis, and in doing so, we get kind of a cell static response. We don't really get an apoptotic response, but we think there's room there to look at combinations in that regard. But the real focus of today was this hypothesis that perhaps in these prostate cancer cells, they can actually take cholesterol that further step through the variety of enzymes that are needed to actually synthesize their own androgens. And so if you were a biochemist, you would love this picture and remember that it's steroidogenesis. For the point of this talk, I'll just try to keep to the key components, and that is to remind you that cholesterol is the beginning component of this pathway. And the key component to make it revert into a androgen or be metabolized into an androgen is that it has to go through either a progesterone-driven pathway or a DHEA pathway to ultimately get down here to testosterone. And then, of course, as I told you, then we convert over to dihydrotestosterone. But in fact, there's been new pathways and new interactions that have been suggested within this, and one of which to point out is that it turns out that cholesterol, if it goes down a progesterone pathway, it can go down what is called the backdoor pathway through a different metabolism profile. And in fact, the key component is that it can ultimately make DHT without making testosterone. So it's kind of a bypass pathway of testosterone altogether. And this has been well shown now in humans as well as a variety of different animals. But the other key take-home message that we'll come back to again and again is I was trying to describe last night, in fact, how this pathway, you almost have to look at it as a city grid and traffic, if you will. And so if you're here on the Granville Street Bridge, here is cholesterol, and you needed to get to Stanley Park, well, there might be a pathway that you prefer, but in fact, if you block off a particular pathway, there's a lot of detours that you could take, and you can cross over with different metabolisms to get down to ultimately making DHT. And that is going to be the real crux of the problem that I present at the end of this. So coming back to, first points of data, I think we and other groups had looked at androgens, and like most people, we've been following these in serum. And in fact, showing here where you have an intact mouse in this case, and you castrate, and you see you get a suppression of DHT, you get a suppression of testosterone along that line. And that's what we've always called castrate levels of androgens, because we happen to look at serum. It wasn't really until people started looking inside the tumors themselves that they saw this factor up here, is that you have a suppression at following castration of testosterone, but in fact in androgen independent or castrate resistant tumors you actually have an elevation of this. And this is even more pronounced in terms of the tissue levels of DHT in those castrate resistant levels. In fact, they're higher than they are in the pre-castrate levels. Another interesting fact, because we were looking across a spectrum of hormones, was we started looking at progesterone as well, and I'll tell you about that later. But one thing to take home at this point is that there's extremely high levels of progesterone in the prostate. And in fact, in the serum, what we find in androgen independence, that there's a lot of progesterone actually being secreted back into that serum. And this really intrigued us as well. But the point is that tumors have a lot of DHT in them in these castrate-resistant diseases. And so we're saying, okay, well, there's hormone there, but we come back to the age-old speculation that it's really coming from adrenal synthesis. But in mice can't actually make androgens from their adrenals. So that was one indication that perhaps it's actually the tumors themselves that are really making these androgens. So we started looking down, in this case showing the pathway of enzymes that are required to get from cholesterol to DHT. And shown in the black bars are the castrate-resistant tumors and the elevation of these enzymes at the mRNA level. This is perhaps more pronounced on the same set of enzymes at the protein level. So there's certainly all the enzymes that we need to make androgens there, and in fact they look like they're elevated, and some of them very statistically and robustly upregulated during this castrate-resistant phase. And on top of that, we started looking at some of these enzymes and recognizing that there's a whole different twist in that some of them appear to be androgen regulated themselves. And so what happens is if you put in a bit of androgens in here, you actually upregulate those androgen regulated in conversion enzymes. And that actually leads then to a feed-forward mechanism. So a little bit of androgens kickstarts the upregulation of the pathway that could ultimately make androgens. So we had really a lot of speculation around this, but to get to the final proof that these tumors could make androgens, we took an approach of metabolic labeling. And to do that, we started with C14 acetate. So acetate is actually upstream of where cholesterol synthesis is made, and ultimately acetate is incorporated not just into fatty acids, but to cholesterol and steroids if they're made in that tissue. So we took tumors that were from non-castrate or intact animals, those that were in this nadir period, and then we took tumors from this castrate-resistant stage, and we wanted to ask, can any of these tumors make steroids, and in particular DHT? And so, in fact, when we run this out on a mass spec, we can see that where this DHT peak is, in fact, these castrate-resistant tumors do definitively make dihydrotestosterone. So that was extremely encouraging along that way. But doing this as a tumor model is far more difficult than trying to actually manipulate cells and culture. So we took the bold step to say, can we actually just take these prostate cancer cells as a cell culture line and induce them to make androgens? Is this an inherent property of what they're doing, and is it really those epithelial cells themselves? And so the short answer to this is yes, we can just take LN-cap cells and culture and incubate those with C14 acetate, and we can make dihydrotestosterone, in the radiolabeled dihydrotestosterone. If we add in androgens to this synthesis protocol, we actually get more DHT being made. So again, kind of enforcing that feed-forward type of loop that once you get the androgen synthesis kick-started, you can make it go. So as I said earlier, we got very interested actually in progesterone. And progesterone is a key metabolite, of course, in this pathway. But progesterone also regulates a number of genes acting through the progesterone receptor, which is very comparable to the androgen receptor, except it activates unique genes. And so we started wondering, what is important about progesterone in terms of it being a substrate for metabolism versus what might it do on its own. And so we can see that the key enzymes that are involved in progesterone synthesis metabolism are all expressed, as I said earlier. Just skip forward here, is that, am I skipping forward or backward? Is if progesterone had an effect on the gene expression of any of these particular enzymes in this pathway. And so the long and short of it is that if we look at these green arrows and red arrows, the top panel, these cells have been treated with progesterone. The bottom panel, they've been treated with dihydrotestosterone, and we're asking if any of these enzymes are androgen or progesterone regulated. In fact, these genes are all changing similarly, whether it's progesterone or androgen, suggesting that they're probably all being changed in expression by androgen synthesis, and the ones at the top are really being changed because progesterone is being metabolized into androgens. However, there were key genes, and this is the particular one, that is solely regulated by progesterone. So this is one of the first observations that, first off, there's a target gene of progesterone through the progesterone receptor in prostate cancer, and it happens to be a really important gene. So it is the gene that regulates the synthesis of, or the bypass of, wherever I am there. It regulates the key synthesis of cholesterol to pregnenolone. And remember, I told you that progesterone levels are quite high. So we had to go through looking at progesterone receptor in prostate cancer tissues, and in fact, progesterone receptor is very highly regulated there. And you look through the literature, and there's virtually nothing known about the molecular actions of progesterone and the progesterone receptor in prostate cancer. And so this interregulation of these pathways got us quite intrigued, and we started wondering, well, what regulates the progesterone receptor and its actions there? And a key piece of information that we got was that androgens suppress, as do progesterone itself, the expression of the progesterone receptor. So again, this time is a negative feedback loop that tries to control ultimately androgen synthesis in this pathway. And so what I'm trying to tell you in this complicated story is that at the point of castration, you have a suppression of androgen receptor activity, but you, at the same time, it kickstarts an upregulation of progesterone receptor activity as well as the presence of progesterone. And during that nadir period, we see that remaining kind of static, and then we ultimately see the upregulation of androgen receptor activity in castrate-resistant disease. And at the same time, because of this negative feedback, we get a suppression of progesterone receptors. So it's a very controlled and very logically put together biological cycle. So what we started looking at then is how different pathways and can work within prostate tumors in terms of making these androgens, and looking at this more specifically, but doing it in a way where I'm just going to skip through that, sorry. In a way that we started saying, well, there's a number of different inhibitors that we have to various different enzymes within this pathway. And of course, the key inhibitor and key enzyme is CYP17A1, which is the target of abiraterone and ketoconazole. And we first off wanted to say, well, is CYP17 actually a protein that's expressed highly in prostate cancer? Because the logic behind that, the development of that, was toward androgen synthesis in the adrenal glands initially. And so it is expressed highly, and it's repressed following androgen ablation. Another key enzyme that we have an inhibitor for, of course, is the 5-alpha reductase inhibitors. And this is data from Jim Muller's group that would say that the 5-alpha reductase inhibitor type 1 is actually quite strongly upregulated in castrate-resistant tissues in particular. And there's also expression of type 2. And this is a key enzyme in this pathway that allows us to further metabolize progesterone into those androgen synthesis pathways. So we took an approach basically to try to dissect this with what happens with various different inhibitors. And the ones that we could pull off the shelf were things like finasteride, ketoconazole, cinnamonic acid, as well as direct androgen receptor antagonists. And we started throwing these enzymes or these inhibitors in to say, how do they affect this synthesis pathway, and can we really block androgen synthesis? And so we went back to those metabolic labeling studies, this time starting with radiolabeled progesterone, and we threw in various different inhibitors. And the point of this is, in a nutshell, is that if we look at PSA levels with different combinations and different inhibitors here, we do affect PSA secretion as an androgen marker differentially. But if we look at the very detailed metabolite protocol patterns that occur from these different inhibitors, we find that they in fact have very different profiles, and I'll try to show that to you in a more defined way here in a second. And that is that if we look to see, in this case, we're asking which steroid is being made in the different combinations of inhibitors, we can see that differentially we affect progesterone versus the next metabolite down, which is 17-hydroxyprogesterone or pregnanolone or DHT. So effectively, these different inhibitors end up with different metabolites down that pathway. And so what does that mean in terms of this pathway? Well, if we throw in finasteride, we can block this, what we call the backdoor pathway, but what we do is end up getting a buildup of progesterone that shunts it down this pathway, in which what we see is we make testosterone in this case, but we also, as we build up this substrate, convert over to make pregnane 3-dione, which then can come down to DHT. So by blocking one enzyme pathway, we force the traffic in this to detour. And similarly, if we put in ketoconazole, which is a CYP17 inhibitor, we get a similar type of problem, is that we may block a particular entity, but we end up, in this case, we're blocking this conversion, but that then forces the steroidogenesis down this pathway. And we ultimately end up with DHT, albeit, and that's why we see different levels of different metabolites coming down that pathway. So there's a number of different inhibitors out there, and we figured, well, why don't we just try to block multiple pathways simultaneously, and surely we can block off all of these detour routes. So we've used combinations of things like abiraterone ketoconazole and dutasteride, and come up with the same type of problem, unfortunately, is that we just keep diverting the pathway. Data's not there for that. But this is clearly giving us at least hope for looking at where there may be particular opportunities to inhibit this intratumoral androgen synthesis in this hormone refractory or castrate resistant disease. And of course, leading the charge on that has been the trials that have come forward with abiraterone. And I think all I have here is the early phase 1 trial, but as you may know, that this is opening into a phase 3 if it's not completely even accrued in its phase 3 trials. But abiraterone specifically does inhibit that CYP17 pathway, and in doing so, it does decrease the intratumoral synthesis, but of androgens. But more importantly, Johann de Bono and his group see a objective response of prostate cancer to this treatment. That's why it's gone forward there. I think Martin can probably discuss more along that way. But our rationale has been, as I said, to try to look at more combinations of various different inhibitors, knowing that any one particular inhibitor is probably going to end up with this diversion pathway problem. And so I think what I'd like to do is really come back up to the top of where we were looking in the beginning, and that is that there are a number of pathways that are activated during prostate cancer progression. Some of these are, in a very coordinated sense, regulated by a transcription factor pathway that regulates lipogenesis. And that lipogenesis pathway itself, ultimately in these prostate cancer cells, can go through cholesterol to create steroids. And that clearly is a well-known cell survival and growth factor for prostate cancer. But we'll continue to explore how these other pathways in their own right of things like cholesterol synthesis and fatty acids synthesis is actually in their own right also promoting cell survival and growth, and whether we can come up with a very clever way with various different pharmacological inhibitors that are already out there to interrupt these pathways all the more. And this work, I really want to state that this is an absolutely key collaborative team effort, in particular with Emma Guns, who I believe you've heard from earlier, as well, of course, Martin Gleave. But the work itself has largely been spearheaded by two really talented graduate students, Jennifer Locke and Amy Lubick, as well as the key technical help from Hans Adamat in the mass spec area, as well as Steve Hendy. And so from there, I'll take questions.